Cool. All right. Hi, everyone. Welcome to a new year of ADSC seminars. It is a great pleasure to have Dr. Michael Lydeamore here from Monash, uh, from the University of Adelaide via Monash, I guess. He is a computational statistician specialising in transmission and control of infectious diseases. And he's worked alongside the Victorian State Government as part of the COVID-19 response, and has more recently focused on healthcare-acquired infections. He has his PhD from the University of Melbourne, but more importantly, a master's from the University of Adelaide, and is now a lecturer in the Department of Econometrics and Business Statistics at Monash. So great pleasure to have him here, kicking off our talks for the year. Take the work.
Thanks, Lewis. And no one told me this was the first one of the year. I might have prepared slightly better. Yes, it's strange being back in this room. I had second-year computer science lectures in here, and lectures weren't recorded in those days, so it was quite a lot fuller back then. But I think of the 200 and something students enrolled, we had a gender balance of 99-1, and it's interesting to see how all of this stuff around universities is changing, although the room has not changed except for the addition of a camera. So I'm going to talk today about two projects in data, health, and biology. And I've got this lovely witty subtitle. It's not the size of your data, it's how you use it. Now, I should point out, Lauren told me this is a very casual seminar, and so I can get away with subtitles like that. And there'll be a few more memes throughout. So a little bit about me. As Lewis said, importantly, I am from the University of Adelaide. I grew up here. I did a Bachelor of Math Sciences and an MPhil here under Andrew, which is why I was commenting earlier that he's heard me talk enough over the years. I moved to Melbourne for my PhD in 2015, and then I joined Monash Uni as a postdoc in the middle of 2019. I did a PhD in infectious diseases modelling and control of outbreaks. I had a position at the government, and then we had an outbreak of an infectious disease that you might have heard of called COVID. So I was recruited into the COVID response in late February, early March. I formed a modeling and analytics team, which later just became the analytics team. And then I rejoined Monash in 2021 after my time in the government was up. And there I joined the Doherty COVID-19 modeling consortium that provided a lot of advice. After all that had settled down a bit, I joined Econometrics and Business Statistics, where I'm a lecturer there in business analytics, which is really data science with a funny name. So the first project I'm going to talk about is the burden of healthcare-associated infections in Australia. And like all things I work in, I did very little of this. This is joint work with Brett Mitchell, Tracy, Bucknell, Alan Cheng, Phil, and Andrew. So what is a healthcare-associated infection? Well, it's an infection that you get when you're having healthcare, sort of what it says on the tin, really. I think you get in hospital. So I think golden staph, MRSA, that's probably the most common healthcare-associated infection. They're associated with varying levels of morbidity and mortality. 5 common ones: C. diff. I can't pronounce what C. diff actually stands for. That causes damage to your colon, can be fatal. Sepsis, bloodstream infections, that has a mortality rate of 15 to 30%. UTIs, these don't tend to kill you, but they, these days, are quite multi-drug resistant, and they also mean you stay significantly longer in hospital, which means you're more likely to get a worse HAI. Healthcare-acquired pneumonia, HAP. This is the most lethal one that we look at, sort of mortality of 40 to 70%. That increases very significantly with age, like all pneumonia. And in fact, ventilator-associated healthcare-acquired pneumonia, so when you're in ICU, they put a tube down your throat and you get pneumonia from that. That's on the 70+% mortality range. And lastly, surgical site infections. So you have a surgical procedure, there's an incision that gets infected. That increases your length of stay a lot, which makes you more prone to these other things. So healthcare-associated infections are not notifiable in Australia. So we rely on the goodwill of hospitals to tell us that they're doing a good job. Most of them do, we think, but we don't really have a robust way to track whether their prevalence is increasing or decreasing. Commonwealth government has this system called a HAC, healthcare-acquired complication. They use diagnosis codes to try and algorithmically work out whether someone's got a HAI or not. There's a fair bit of research out there now that says that those underestimate by somewhere between 15 and 30%, and not even in a nice random way where we can just pump all the numbers up. So overseas, the situation's not quite so grim. They are actively monitored across Europe. The European Centre for Disease Control do a good job of this. And in 2016, the ECDC published an estimate of the annual burden based on a survey in 2012 data of just over 2.6 million new HAIs that year. So the data for this was a point prevalence survey, and I'll talk about what those are in a second. And this is a very large one by PPS standards, 273,000 odd patients and just over 1,000 hospitals. So we're collecting a lot of information there from patients. So a point prevalence survey sort of does what it says on the tin. It's the prevalence at a point in time of a condition. Okay, so the way these practically work is that we get a bunch of a research team, clinical nurses, a clinician, and a team of research associates go out to a whole bunch of hospitals. They look through every patient that's in there on a given day, and they follow a flow chart to decide whether or not this patient has a healthcare associated infection or not, and if they do have it, when they got it. So we ran a PPS in 2019, and it consisted of adults in hospitals. That's a good start. I mean, really, it's large hospitals. We didn't go to the small ones. There's a lot of small hospitals in Australia. Actually, only the large public ones, because the private ones wouldn't let us in. And it was only the acute care ones of those, because the smaller ones are pretty hard to sample. Actually, it was only 19 large acute care public hospitals. Okay, and for context, Victoria alone has 80 public hospitals. Nationwide in Australia, we have probably about 200. Nonetheless, this does actually make up a fairly large percentage of the hospital admissions in Australia, about 60% of them. So we're getting reasonable coverage, although caveat that because we're only sampling large acute care public hospitals, we're going to miss things that are happening in day surgeries. We're going to miss things that are happening in children. We're going to miss things that are happening in hematology centres and things where people go for short stays. So all the acute care wards were included. Non-acute wards, so that's things like emergency short stay units, pediatrics, neonatal intensive care units, because they're children, rehab wards, and the emergency department are all excluded. The pediatrics and the NICUs are because we were only sampling adults. The rest of those are because your chance of picking up an infection in those short stay places is very small, and they have a colossal volume of patients. So there's a cost trade-off there. So as I said, these hospitals make up about 60% of the overnight admissions to Australia. Okay, if you include same-day admissions, that number's way smaller. Your chance of picking up an infection in a same-day admission is quite small, so we're sort of all right with that. And we're not doing another one anyway, so we sort of have to be all right with that. Okay, so we sampled 2,767 patients. Australia is quite big and money is quite small, so we actually sampled these over a few months. Actually, it was 2 research associates and 1 clinical nurse, toured around the country, stopped in all the different hospitals. And on any given day, they would go in and sample all the patients, or every second patient was in there. We cover a pretty wide range, including one 104-year-old, a median age of 67. That's about what you expect for overnight admissions. Roughly even gender balance. And almost all the patients are in major city hospitals, but then again, all the hospitals are in major cities, so we're probably about on par there. It's methodology time, right? I haven't given a presentation in the maths department for a long time. And so I've put some maths in so that we all feel a bit better. Our task is to go from point prevalence, which is what the data measures, to annual incidence, which is what people actually care about. Okay, so step 1, we're going to go from, we're going to estimate the hospital prevalence. Okay, remember, we're not measuring every patient in a hospital, so we've got to extrapolate that up. And we do that through this mixture of beta distributions where someone more intelligent than me has told me this is a standard thing to do. So we extrapolate from a binomial sample with lots of zeros, which is why we need the mixture. These conditions, although I'm standing up here like they're really common, are actually not. And so you have a lot of patients that don't have anything. Amongst our 2,700 odd patients, I think about 200 experience AHAI. Okay, so we have our number of observed patients and then our number of patients in the PPS. You take a mixture of those with a fairly flat uniform prior on the R, and you get some estimate of hospital prevalence out. Lauren's looking at me quizzically, but I'm sure it's fine. Dan Simpson told me it was fine. That's probably right. Right, now we're going to go from hospital prevalence to hospital incidence. Okay, so incidence is like the amount of cases over a period of time. So if you actually reverse this expression, it's slightly easier to think about. Okay, so number of infected people times the length of infection, that's going to give you the total number of infections over an infectious period. Divide that by the length of stay, and that'll give you the prevalence at a point in time. Flip all that around, and you get this relatively straightforward term. So P we already measured, that's step one. So all we need now is the length of stay and the length of infection. We actually capture the length of stay through the Australian Institute of Hospitals and Welfare, Health and Welfare, AIHW. And that has some party emojis on it because not every country does that, including, it's not a country, but the economic region of Europe. Length of infection, though, that's not available. And the reason that's not available is we only go to these hospitals once. So all we see is what I'm denoting LOI PPS, right? So how long has a patient had their infection up to the day that I'm there? Okay, so you can empirically calculate the probability that someone is on their first day of infection. And then it turns out that if you make some fairly strong but reasonable assumptions about what that distribution of infection looks like, the inverse of that gives you a surprisingly good estimate of the mean. Okay, it's called an exponential distribution. We've probably heard of it. For a small sample size, it turns out some ECDC research says that's a horrendous estimate of the length of infection. And we have a small sample size. So in reality, what we actually do is take a mixture of that term and the empirical mean. Okay, so same way we were taking the mixture of the two distributions before, another mixture. That helps get rid of our bias, but it does increase our uncertainty a lot, as you will see on some plots. Okay, so now we've got hospital incidents. So now we can convert that to population incidents, and that's the easiest step. Multiply it by the number of discharges. And we capture that too. Yay. Okay, there's the magic number. It's 60% of the total admissions in a given year. Okay, IHW reports that number. And interestingly, again, Europe doesn't capture the number of admissions they have. And so they have to resort to a slightly egregious estimator using the number of patient days and the number of patients, and you can undo those and get some estimate that's pretty good. Okay, so if you were wondering, multiply or divide that number by 0.6, and that's how many overnight hospital admissions there are in Australia per year. The overnight is important. We probably have double that on their own of same-day admissions. Okay, step 4 is to stratify by age and sex. Lauren, just block your ears while we stratify something. We'll use a multinomial likelihood. We use a Dirichlet prior. That gets you some nice conjugate prior arguments. We use weights taken from the number of cases in each age sex category. Caveat, these things are rare. Our sample is small. You need non-zero counts in every bin. We don't have that. And so the accepted but incorrect methodological solution is to add this small pseudo count to every bin so that it's non-empty. Okay, and Lauren has told me just because it's called W doesn't make it a weight. And that's true. But today it is a weight and we'll survive. Okay, the last thing we're going to do is look at deaths. Okay, we like to measure mortality, so we'll adjust for life expectancy using this thing called a McCabe score. Another typo. We'll use that. So a McCabe score is a qualitative assessment of how likely a patient is to die. Fairly morbid thing to do. Patients can be categorized as non-fatal. We like that one. Fatal, which has an average life expectancy of 3 years. Or the worst category, rapidly fatal. You don't want to be rapidly fatal. That has an average life expectancy of half a year. In practice, to do this, to do it properly, you have to have a clinician look at each set of case notes and make that judgment. Clinicians are really expensive. So a lot of surveys, including our one, don't collect McCabe scores. So what we will do is take the distribution of McCabe scores for the various conditions by age and sex and just apply them to our data set. Okay, we have a pretty similar population structure to where most of this data was collected, so it's probably fine, but we don't actually know in practice. We combine these with things called a disease outcome tree, and then we can calculate disability adjusted life years, which is this measure of how bad a condition is to try and let you compare across things. and also mortality. There's supposed to be a picture of a disease outcome tree there. Well, no picture for you. I can draw one. No one online will be able to see it. Right? It's a tree, right? So you have bloodstream infections. They can either be complicated, right, or uncomplicated. Complicated is about 20, uncomplicated is about 80%, right? And then you get things from complicated bloodstream infections. So it could be PTSD is a common one from BSI. You can develop sepsis, right? Uncomplicated tends to have recovery only in most conditions. Each of these things is well known and has some disability weight, right? So a disability weight of 1 means you're dead, very disabled, about as disabled as you can get. 0 means no impact. So PTSD from memory has a weight of about 0.05, okay, which sounds small, but you have it for the rest of your life. So it adds up to quite a lot. Sepsis, right, has a disability weight of about 0.8, but it tends to kill you. So when you're doing disability life years, you sort of only calculate it to the point of death, and then you have to take mortality after that. Right, so people think a lot about how these things work out, and then clinicians try and work out how to get a tree out of it. The ECDC's done almost all the work for us here, and they provide disease outcome trees for a huge amount of conditions. So this is normally a public health talk, and it's customary to have a table of too many numbers in a public health talk. I haven't highlighted all the p-values that are less than 0.05. That would be the other convention in public health. Nonetheless, the takeaway number is very slightly darker down here. Okay, 170,574 healthcare-associated infections per year. And if I put that into some numbers that make sense, that's 1 in 20 admissions resulting in an avoidable infection. And they are largely preventable healthcare-associated infections. 7,500 deaths and 122,000 dailies. I'll put that in some context later as well. So that's a pretty big burden of disease for a thing that we don't capture, don't really try and control, and don't really try and prevent either. So we can compare this. This is normalized per 100,000 population. We've got females to the left, males to the right, Australia. So the estimates you just saw in these grey boxes, the ECDC estimates are in orange, and what's called the German convenience sample, which I think makes it sound like a supermarket. It's actually just some hospital data they happen to have lying around, is in blue. Okay, and so the left one here is the number of infections, the right one is the disability adjusted life years. So the first thing to notice, the plot goes a lot further to the left, which is that way, to the right. Okay, so we have a female bias here. We don't see that nearly as much overseas. I have probed some clinicians and infection prevention people for why that's the case. No one seems to really be aware that that could be occurring, but it's a pretty consistent trend across all of our different conditions. We have this sort of funnel shape, so it gets worse as you get older. As I get older, I can almost guarantee that's the case. And I assume that only gets worse the older you get. So older people get more things, they're in hospital more, perhaps that's also expected. Worth noting, not that I think anyone will, but if you ever go to try and pull out the original version of this plot from the CDC paper, the age bins are completely different. So I've had to rebin this and re-estimate it basically, but they capture all the way down to age 0, and they have so much more they can do this by 5-year band. Because our survey's smaller, the uncertainty is also a lot bigger, which is why the whiskers are so wide. So this is broken down by each condition. Now, I'm not expecting you to be able to look at this and immediately tell me everything about it. But if we go down to bloodstream infections here in bottom right, this is just how big our grey bars are. And it's fairly similar to healthcare-associated pneumonia. So what does it say? We have a colossal amount of bloodstream infections in Australian hospitals. Again, I don't think we really know why we have so many more. Pneumonia, I think we have some idea. We're a bit ventilator happy in Australia. So hospitals are often very keen to put people on ventilators. That does come with other problems like this. Bloodstream infections, I mean, there's so many of them that the first time I looked at this, I was like, ooh, I've made an error here. But if you look at the empirical rates out of the survey, it does sort of line up. So there's currently 2 spin-off projects from this one. One looking at what on earth's going on with bloodstream infections in a couple of hospitals. and one looking at intensive care, specifically at ventilator-associated pneumonia, which is even harder to measure.
Is there a preference to, like, is that why we see such a huge skew?
Maybe
Is that where the trigger happiness is coming in?
it could be. I don't know. Some of the up here, some of this is because generally there's more women, older women in hospital. So even controlling per population is probably not quite the right denominator. You probably want to control per admitted population. But it could be. Yeah, don't know.
The bloodstream infections, is that cervical wounds infections?
Primarily, yeah, not always, but often. So, but even if you went in hospital with some sort of skin infection, developed skin sores from that, and those got infected, that would only if they operated on them, that would count as a surgical site. And
It's captured into
maternity data, yes, yes. So I believe there was no exclusion of those hospitals. So yes, it should be in there. So here's the attributable mortality. You can do this by condition. The numbers get quite small, so I've combined them all together here. It's a large number. It's larger than overseas, certainly in the older population. Yes.
Do you think that hospitals for the same reasons as folks? Like it's the stats.
I don't think anyone's ever looked at that. That sounds like it will be a data nightmare because other countries basically never want to share data overseas. So you'd have to either compare across studies or have a very good collaborative network that has enough links to get the data. But no, I don't know the answer to that. Okay, so some novelty. This is the first estimate of the healthcare associated burden ever in Australia using a point prevalence survey. There have been estimates from single hospitals or single studies that have done a reasonable job of trying to extrapolate that out to a whole population. This is the first time we've been able to do it Australia-wide. Part of that is because we don't do point prevalence surveys in Australia very often. So this 2017 survey was the first one since 1984. We are in the process of doing another one, but there's still no established routine system for doing this. It's still based on whoever wants to fund it at a given time. So we have no routine surveillance of these things, but that means that these surveys are still the only way to robustly understand the full burden. So I said I contextualize these DALYs, right? So 498 DALYs per 100,000. If we compare that to motor vehicles, it's way worse. Infectious diseases, this was a pre-COVID number, so that may no longer be the right amount of DALYs. But obviously it's not as much as respiratory diseases, which have an enormous burden. Things like influenza and even rhinovirus cause small amounts of disability, but to a lot of people for a long time. Nonetheless, this is putting it right in the middle of things that public health decision makers think are very important. And so really, I think the takeaway from this, which we'll get into a minute, this did make quite a stir when we published it, and there's now new guidance on how to do this surveillance on healthcare-associated infections in Australia. There's a dedicated MRFF scheme based on this number that is designed to better understand how healthcare-associated infections work. And this is all based on just 2,767 patients from only 19 hospitals. And so in the scheme of modern data, that's very small. So, as I said, these HAIs are preventable, and they represent a huge opportunity for improvement, but we do have a long way to go to get anywhere near preventing them entirely. So I'm going to pivot now to another project. So to make life a bit easier, does anyone want to ask anything about this one? Yes.
So you mentioned you couldn't get the McCabe scores for patients like that. Now, I know for other scorecard type metrics that are applied in healthcare, typically the nurses can actually guess the scorecard before they've gone through the groups.
Mm-hmm.
Is there an equivalent that could be used?
I suspect that's true for McCabe score. As you probably know, a lot of clinical researchers love a protocol, and the protocol says a clinician has to do it. So I would almost guarantee you don't actually need a clinician to do it. Nonetheless, that's all that's ever looked at. It's a decent idea though to see how well that goes. Certainly for the training of the data collectors, they do that sort of thing. So someone comes and trains the data collectors and then validates them a few times, and then you can just use more untrained collectors. Yeah. Any other questions? Is my chat clear? Great. Let's go totally the other way. We're going to move away from healthcare, and we're going to look at something that happens to a lot of single cell data. Okay, so you might have a new drug, you want to know what it does. You say, give the mouse some of this drug. You then take some blood from that mouse and you do what's called single cell transcriptomics or genomics. Basically count the different types of cells, all the genes they express. And then you say, look, these genes react with this compound. That must mean new drug is good. To do that, use something often called nonlinear dimension reduction. Okay, so let's start with high dimensional data. The world collects a lot of data. I think we all know that in this audience. Much of this data is very wide. And by wide, I mean high dimensional, right? You might collect 10,000 features on a given cell. That would be a pretty standard amount. It's very challenging to look in 10,000D. We can look in 2D. We can pretend we can look in 3D, although we're not very good at it. So as I was talking about with single cell RNA data, they're looking for these groups of cells with similar expression profiles and looking for these marker genes. But how can we check that whatever we're doing to this data to identify that what we have is a real feature is real and has not been introduced by any of these dimension reduction processes or model processes or clustering or anything like that? Well, one thing we can do is look at this thing called a tour. So this is a tour. This is a 7-dimensional S with a little bit of noise added to it. Okay, so if you see as it pivots around in a few different angles, you'll see it looks like an S. There you go, there's an S. Okay, for certain views, it looks flat. These are interactive. Okay, so I can actually come here and I can highlight certain points. I can fix an axis if I like. X7 is a bad one to fix. Let's try X1. There you go. You can have it pivot around there. They're a way to look at, in theory, infinitely high, but finite, as large as you like, but finitely dimensioned data on a 2D view. Okay. Strictly speaking, what's happening here is you're seeing a sequence of linear projections and some sort of mapping between them. There's various ways to generate these different projections. Some of them try to maximize variance. Some of them try to minimize distance between the views. All of them end up with some sort of scrolling image that looks a bit like this. Tours aren't used all the time. Okay, they're a little bit hard to think about. That's a particularly nice example, right? Because it's clearly an S in high dimensions. You can imagine if I've got a blob with a bit sticking out, I might have to stare at it for 10 minutes before I see the blob coming out. It's not great. So one common thing that people do is dimension reduction, right? And I learned about dimension reduction as an undergrad. I learned what PCA was. That's a dimension reduction technique. Okay, that's a linear mapping. Has to be linear. It's a product of matrices. Linear mappings are great. We use PCA all the time, but they don't always capture everything. You might have nonlinear interactions, in which case you might want to use a nonlinear dimension reduction technique. Typically, these are designed to maximize your variance in your 2D view. The claims of these algorithms is they do that while also preserving pairwise distance from my high dimension to my low dimension. Okay, so some of these popular ones, at least in bioland, UMAP, TSNE, PACMAP, FATE is a new one that's very popular. Okay, lots of these different nonlinear dimension reduction algorithms. But as with anything that's designed to maximize variance, they can invent structure or make structure look far more clear than it actually is. Okay, so a canonical example of this is if I draw 10-dimensional balls in Gaussian balls and then put them quite close together, flatten them with UMAP, they appear all around the edge of my plot and they look really far apart. So we can specify dimension reduction formally. Okay, our high D data will be rectangular, x n by p, so n observations, we'll carry that all the way through, p dimensions, so p is my high D dimension. And then we're looking for a projection, same number of observations n, but D, where D is much smaller than P. Straightforward enough. Okay, here's an example of some nonlinear dimension reduction. So this goes UMAP, TSNE, FATE, PACMAP, and I forgot the name of the last one. There are 5 different NLDR techniques, all on that S-curve data that we just looked at in the tour. And they look similar-ish, right? Like I could probably guess that all of those would come from various views of that S-curve if I saw the S-curve with it. Okay, but particularly plot A in the red and plot D have quite different looking pictures. And actually it gets even worse than that, because this is just UMAP 4 times with different parameterizations of the algorithm. And now I've got a very sort of piecemeal structure all the way down to what looks like an almost sort of smooth surface going around. So there's clearly some need to validate what these models are doing in our higher dimension. So we, and for a very loose definition of we, it's almost entirely Jan Ilachika, who's one of my PhD students who's working on this, have developed a methodology, an algorithm to project those 2D dimension reductions back up into higher dimensions again. So we can effectively look at model fit through a tour. Okay, so we're going to go through 4 steps. 1, partition our dimension reduction data into hexagonal bins. Okay, nice way to bin up our data in 2D that tiles well. We're going to triangulate the centroids of these bins to form a 2D manifold. That's probably a manifold. I don't think anyone in here is quite precise enough to tell me if it's not a manifold. And then we'll map these 2D bins back up to high D using the points within each bin. And then we'll measure model error and visualize in high D. Okay, so let's step through this a bit. Here's our S-curve that's been through UMAP. Okay, and then we just do standard hex gridding. Okay. Algorithms have been around for this since the 70s. Lay out a grid of hexagons of a specified size. We're forcing them all to be regular hexagons. It just makes the map a bit nicer. It's not actually a requirement. And then count how many are in each cell. Okay, I'm petitioning for this to be called hexagonate because it sounds like triangulate and it's a good word. I am losing that battle. And then we just turn the left one and the right one, plot our centroids of each hex. Okay, now there's a little step 1A in here that I'm not going to talk about much today, which is what to do if we have like this bin here that's only got 1 point in it. Should you include that with the same importance as all the other bins? This software actually comes with an option to choose that threshold to remove what we are calling low density bins. Okay, then we triangulate them. We're going to use linear triangulation, creates a triangular mesh. Okay, it connects to its 2 nearest neighbors. Because our hexagons are regular, most of the triangles, at least if they're in adjacent bins, will also be equilateral. And it's not actually a requirement, it's just nice. It's a bit easier to look at. So you can see like down here, where the majority of our data was, we've got this sort of nice equilateral mesh looking thing. And then a handful of these really ugly long edges going all the way along the outside. This long edges is more of a algorithmic artifact than a data artifact. And so we also provide the option to remove long edges. There's a system for doing this. You look at the largest gap between points and then you sort of slice it there and then you can look at the result. So the red edges here are all classified as long, including that one that's right on the threshold. Okay, and the resulting surface will just be these black lines. So now it's time to map from 2D to high D. And this is definitely the most maths I've put in a presentation for a long time. So bear with me. We're going to define a function f that goes from Rp, so that's our high dimensional space, down to R2. So that's mapping its high D point to where it is in NLDR space. Then the set of all points in a single hexagon, which I'll label by I, will denote that capital HI with a centroid little hi. Now we need another function, okay, 2D to 2D. That's going to map the 2D point to its centroid. Okay, so whichever hex it's in, it's pushing it to the middle. So then compose those together, you get a function that maps the high D point to its UMapped centroid. So we take all the points in our centroid, they're all labeled now by I. We calculate their high dimensional mean. Okay, so 1 divided by the number of points, sum them all up, gives a high D mean. This D is a slightly different D to the previous D. Don't worry about it, doesn't matter. It's now a high dimensional D. We won't go back to the low D one. Then we choose a function V that goes from low D to high D that looks like this. So that is V maps the 2D centroid to the high D mean of the points in that hexagon. So if everything's working well, right? Remember, pairwise distances are preserved. So points that are close to each other should be in the same hex. Thus, they should be close to each other back up in high dimensions. So this function gives us a way to say, here's a bunch of points that are close together after we do an LDR. If we put that point back up in high D, are they all close to each other still? Okay, so you can write that out, right? So F composed with G gives the 2D centroid associated with a point. V of F of G of X gives the high D centroid associated with the 2D embedding of point X, which is really what we're after. So we'll use this to define a model of the 2D embedding back up in high D, and then we can look at it using a tour, and you can even compute error and AIC and MSE and really whatever metrics you like. So here's what it looks like, right? This is the same S-curve as before, but I've put that mesh after we've removed the long edges on it. That looks pretty good. Right, the dots are roughly where the lines are. It's nice. Interestingly, all of those UMAP projections that I showed you, those 4 different ones, give you pretty much this mesh. And the reason for that is that space, when you dimension reduce, doesn't mean space anymore, right? Like you can twist and flip, you can't squash. And they all mean the same thing. So just because I've got one curve that looks like a upside down U and one curve that looks like a sort of slightly stretched C, if I rotate that around, they mean the same thing, so it projects back up nicely.
You mean it literally is the same topology?
Yeah. So simulated world is nice. We have done this on a real world example. This is the single cell data. So the left figure comes from one of the published papers in single cell land. This is a common example data set that people use. So there's 10 different cell types indicated by the colors. 10, 1, 2, 3, 4. Yeah, 10. The way that this is worked out practically, the data gets put through UMAP, then they use k-means clustering. They expect 10 different cell types. They ask for 10 clusters. This is the answer you get back. Now, we all know that if you ask k-means for some number of clusters, you will always get that number back. Right, first time I look at this, you sort of think, wow, this is a strong line to draw in this cluster, maybe the same thing up here. Nonetheless, you do our 2D projection, and then this is a visualization of the error that's really hard to look at. I have got this in tour space. So this is the high dimensional data after PCA. Okay, remember we're looking for 10 clusters here. Perhaps we're more looking for 3 clusters, right? 1, 2, 3. They look really far apart in UMAP view, remember? Well, there was a little bit of separation. It's there a little bit. Perhaps not as strongly as dimension reduction with success. I'll put the model on over the top. Okay, so there's a few key points. Now it's really hard to see, and I can pause this. I'll try and see when it comes up. Oh, there's a good one. Right here. These 2 points are really close together in high D space, but they're really far apart in UMAP space. And that's what's giving us those 3 distinct clusters. And so what you can actually do, as my phone's telling me that I'm running out of time, so I'll pick up the pace slightly, is because we can calculate an error, you can do different parameterizations of UMAP and see if you can get a better high D model fit. And if you do that, you end up with this. This is still UMAP. I've just changed what parameters go into UMAP. And now I'm still asking for 10 clusters. And I've taken the labels off of the previous one, so I'm cheating a little bit here with this labeling process. But I get a smoosh of CD stars, right? CD4s are close to each other. Here's the CD8s. I get some other bits and pieces, but it's much more connected. Right, and I don't think I have a tour of this one. And it's really hard to see on this projector especially, but the error here overall is lower. Okay, so this is a semi-automated way to tune your dimension reduction that can preserve your high D distance. Now I say semi because in theory, adding a bin will always reduce your error. And so the canonical answer to this is to have as many bins as you do data points. It's not very useful. It's just like choosing a histogram size, really. Okay, but it helps give us some tools to validate these things in high dimensions. Okay, so to sum this up, dimension reduction is really hard. Maximizing variance can create structure or elongate structures that perhaps aren't as strong as they look. Visualizing in high Ds using a tour is a great idea, and we should do it in lots of things more and more. But it works particularly well here to actually see if our dimension reduction works. And the algorithm and R package associated with it helps create easier visualizations for end users who, at the end of the day, are doing the best they can without understanding all the details of NLDR like we all do all the time. Okay, so just to say thanks to Gianni, who's the PhD student who's done all of this work, including all the software development that goes with it. So I just want to finish up, I guess, taking us back to the title. Right? Don't let a small amount of data limit your impact. Okay, data is expensive. We all want more of it all the time. We'd love more, right? The first time I met Lauren, she was telling me how she really wanted some more data. And then it was, it's hard to get more data on these things. A handful of observations leads to real change. The flip side of this is that more data doesn't mean more good. Okay, and you can't model your way out of bad data, but you can model good data badly. And so I'm now working on both ends of this spectrum, which I think is really fun. But I guess my sort of life motto now is that these impactful projects really rely on good people, good problems, and good techniques far more than the amount of data that goes with it. So I'll finish up there. Happy to answer any questions. Thanks for coming. Yeah.
About that dimension reduction, right? Because you said that the same topology gives different pitches depending on the view you see it at, right? So you just for like clusters, are you just looking for you just basically like choose different parameter values and just see whether there's any sort of segregation? Is that what you're picking? It's determining? Are you like, is that almost picking the ideal parameters that give you the picture of segregation you see?
Yes, but it's the segregation in high dimensions, not in low dimensions. So what we're doing is choosing parameters for an algorithm. You could also choose an algorithm if you want to treat that as a parameter. That makes this picture look as similar as possible to the high D picture.
Yeah.
So the fact that there are clusters on our real world example here doesn't actually play into the algorithm. So the algorithm just sees these as all points. When I was talking about separation between the clusters, I was talking about this here, right? So you see these three, and then you look in the tour and they don't look that separate. Whereas if you choose based on the error metric that Gianni developed, you get a much more connected picture, although you still can get your 10 separated clusters, which is what the biologists really want because they expect 10 types of cells, they should be able to see them.
Yeah.
Does that answer that question?
Yes.
Yeah? Lewis?
I mean, those points that are on the boundary, the hexagon, are more likely to be put into the role.
Yeah.
So therefore doesn't choice the hexagon.
Yes, it does. Yeah. So the software has a parameter in it where you can offset the grid by up to one full hexagon in either direction or both at the same time. You could put that into the optimization procedure as well to try and avoid that. I have thought about what wrapping means. So if I have a point on the edge of a UMAP, two edges of a UMAP, they're never going to be connected. But it's unclear to me whether that should effectively be a periodic boundary or not. That sort of thing. It's a good question. Lauren?
Is your error rate, is that by hexagon or is that pointwise?
So the mean squared error is pointwise. But if you plug it into an AIC where you need a number of parameters, then the number of non-empty bins is what is the number of parameters. So that means then, because there's no number of bins in the MSE, the error cannot increase by adding another hexagon. So the MSE is sort of a terrible measure for optimizing. The AIC does eventually bounce back up again when you're just adding pointless bins. You could do it bin-wise. We did think about that. I can't remember why that didn't work that well, but there was a reason.
Can I ask a tour question, because it's been bugging me since I ever first saw it? How high have you gone with dimensions in that to be able to visualize? Like, could you put in 380 and still see something?
I don't actually see the structure, I'm just saying the software works.
Yeah.
So this particular project, I don't think has gone higher than 10D. Guy Cook would know the answer to that, as to how high you can go and still see some information.
Sort of went up to 4, so I was really
Yeah.
interested to see 7 in your talk and wondered.
Yeah, so the next step actually of Jani's PhD is to do a visual inference experiment. So put a dimension reduction next to a tour and then ask people, are these the same thing or not? And one of the factors in that experiment is the number of noise dimensions. And I think we're going all the way up to 10 for that. But yeah, Di would definitely know the answer to how many dimensions can you realistically see on a tour?
You know, I mean, on that, we started off by saying here's a 7-dimensional S.
It's not really a
But it's
7-dimensional.
not, it's 3D, and you, then there are random.
It's 3D with some noise. Yes, that's right.
There's a question from the chat. What are the dimensions in this real world example?
As in how many? Yeah. 15. But it's been through PCA before this has been done, so that just standardizes all of it. So I think they collect over 100 in the actual original data, and then it's been standardized in inverted commas using PCA.
There's a follow-up question. What are the names? But I've got to skedaddle, so I think.
Yeah, I can manage the chat briefly. What do the dimensions mean? Well, nothing when they're PCA'd. You can look for what goes into each PCA and then say what original things end up in that PCA column, like you would do for a standard PCA variance analysis. In this case, they would all be amount of gene expressed by a cell. Any other questions?
All right, what a perfect talk illustrating the concept of data science here. It's somewhere between data, statistics, and mathematical modeling. So let's think about it. If anyone wants to go for lunch, we'll go for lunch. Sure.
I can't stop the recording.
And
Oh, I can end it for everyone. Yes.